Hello, everybody. I'm told that we are live, so welcome to Creation Myths. I'm just going to refresh on YouTube to make sure that we are actually live before I go any further, including introducing my guest. We look good. So, hi, everybody. I'm Dan. I'm your host, and joining me today for the second time returning is Dr. Michael Behe. He is a professor of biological sciences at Lehigh University. He's the author of Darwin's Black Box, The Edge of Evolution, Darwin Devolves, and most recently, A Mousetrap for Darwinism. He's a senior fellow at the Discovery Institute Center for Science and Culture. And I forgot to mention this last time, but it's important. One of the star witnesses at the Kitzmiller v. Dover Intelligent Design Trial, which was in 2005. And fun story for both you, Dr. Behe, and for everybody else, that was the thing that got me into evolution and creationism as intelligent design. Was following that trial in real time as it happened was like what got me into evolutionary biology. So welcome back. Did I miss anything in terms of your introduction?
No, that does it. If anybody wants more information, they can look at the Wikipedia page on me.
There you go. So before we go any further, I really want to just thank Dr. Behe for being here. It's hard to get engagement in this conversation with kind of professionals that disagree with the evolutionary consensus. This is the second time Dr. Behe has come to talk with me. So especially after our first conversation, which I disagreed with a lot of what he wrote in Darwin's Black Box, I really appreciate you coming back on for a second conversation. So thank you for being here.
Yeah, you bet. You're not alone in disagreeing with me, so don't feel shy about it.
So the first thing where I want to start is what we're going to talk about today is your second book, The Edge of Evolution. And I want to give you the opportunity to just take a few minutes to kind of summarize the broad arguments you make in that book. Before we go any further, I've now read the book. I just reread the book, so I've now read it twice. I'm sure most of the audience hasn't read it. So give us the 10,000-foot view.
Okay, here's the scoop. In my first book, Darwin's Black Box, I pointed to a lot of biochemical machinery that I said was irreducibly complex and could not, or was a very poor candidate for being explained by Darwin's theory. So there are some things in life, I said, that Darwin's theory can't account for, but I think it can account for some things, certainly. The shape of my nose might, not be irreducibly complex. And I thought there were a number of things that showed that, yes, random mutation and selection can explain things. So if you're like me, you think there are some things that can't, but some things that can. A question that occurs naturally is, well, where's the broad dividing line between those two? And I called that the edge of evolution because it's alliterative, and that's what helps titles. And the main example in my book, I tried to look for experiments or field observations that showed how much evolution could occur and what it could do. And just around then, the molecular mechanism behind chloroquine resistance in malaria was published and became known. And it turns out that malaria exist in enormous numbers on Earth, and they infect lots and lots of people. And because of their effects on human health, a lot is known about their genetics and so on. And I wrote that chloroquine, which was the drug of choice for malaria, lasted so long, that is, before resistance to it evolved in the wild, because it likely required 2 mutations before it was the resistance mechanism was effective. Not just 1, but 2. And I laid out my reasons for thinking that. And then I went on and tried to generalize that to use it as a rule for deciding what, in a much broader scheme, Darwin's theory could account for and what it can't. And I decided that, or I argued that, that if some new feature required 2 novel protein-protein binding sites, then that would be beyond the Darwinian capabilities. And I won't go into that. We can go in during the discussion if we want to. But in general, the problem for Darwin's theory is what he outlined back in The Origin of Species. He said at one point, if it could be shown that any organ existed which could not be reached by numerous successive slight modifications, his theory would break down. And he insisted that each step had to be selected, or pretty close to each step. And it turns out that Darwinian magic goes away, it doesn't work, if one step in an evolutionary pathway or a couple steps are not selected. Either they're neutral or they're unfavorable, they're degradatory. And so in these examples, I tried to argue that that was a good observation of Darwin's, and it looks like there are such things that he didn't know about.
Yeah, okay. So exactly. So you spend a lot of time in the book looking at Plasmodium and the interaction between Plasmodium and humans. Look at HIV. You spend a little bit of time looking at E. coli and the Lenski long-term evolution experiment. If I could summarize, I want to make sure I have it so we're not operating from kind of two different views here. So the idea is to look at organisms where we know the molecular evolution for a specific trait. We can look at a specific trait. We say, okay, these are the specific mutations that cause it. We have enormous population sizes, orders of magnitude beyond human population sizes. So we know we're catching as broad a swath of the possible mutations in those genomes as possible. And for many of the cases, it's every possible mutation what we're surveying. So you have big populations, lots of mutations in a relatively short amount of time because you have such a big population. And the idea is you're establishing the maximum rate at which evolutionary processes can generate novel traits that require 2 or 3 or 6 or however many specific mutations. And the emphasis is really on that rate, and then looking at other traits, like, forget drug resistance, just looking at like, okay, well, drug resistance happens at this rate. Look at this other trait that requires more complexity. If drug resistance happens at a certain rate, and we can establish that rate, then these more complex traits would happen too slowly for evolution to be able to account for them. Is that a fair characterization of the argument?
Yeah, that's very good. I would just say one thing, and it's not just the rate, but as you know, rate is really kind of tied up with population size, because with population size, you've got a lot more opportunities for mutations to arise. But those two together, yes, that's right.
Gotcha. Cool. Okay, great. So for today's conversation, I know you've talked about this a lot, both in person, like this, and then also in writing at length. People have talked about back and forth with the malaria examples, the HIV examples. I know there's a long blog exchange from back in the blog days with Larry Moran. There was the long exchange about HIV with Abbie Smith and Ian Musgrave. I don't want to rehash any of that today, so don't worry. We're not going to get into the specifics of like many mutations. We're not going to do that. So what I want to start with is what I took out of this book for what we agree on, which is a lot, actually. So Earth and life are billions of years old. I'm going to say cellular life, because viruses are weird. But I'm going to say all cellular life shares universal common descent, and there's extremely strong evidence for that, especially at the molecular level. So the question where you and I disagree is the how. It's what processes explain how you get from A to B to C all the way down to all the Z's that exist, in the modern world, right? So that's where we butt heads when it comes to explaining all that diversity from a universal common ancestor. Is that fair? I haven't attributed any to you that you don't hold. Cool.
Yes, that sounds good.
So now we're getting into it. So now what I really want to talk about this book is the underlying evolutionary biology, because as I'm reading this book as an evolutionary biologist, and I got a lot of problems with the underlying evolutionary theory that's presented in this book. So I'm going to start with a bunch of small scale problems. I don't want to call them small problems because they're pretty big problems, but they're small scale, and I don't want to spend a lot of time on them because there's a couple big themes that I would really love to pick your brain about. But just a couple small things that jumped out at me. Like throughout the book, you use Darwinism as a stand-in for evolution. And we talked about this last time a bit. Like, Darwinists don't exist anymore. Nobody thinks Darwinian evolution explains everything anymore. We're way past that. And again, we talked about this last time, so we need to harp on it. It's just like when I read a book about Darwinism, it's just like nails on a chalkboard because nobody talks like that anymore. In chapter 1, there was the incorrect definition of fitness, just definition of evolutionary fitness was off. Chapter 4, there was a very small bit, just a description of mitochondrial Eve, and it wasn't quite right for like what that means in terms of being the mitochondrial most recent common ancestor. There was the implication that Darwinian evolution or evolution in general or selection specifically, the sentence isn't quite clear, but there was the implication that it's random. It's not random, but it was set up kind of as evolution in contrast to non-random process. Like, well, evolution is non-random. It's just parts of it are randomish. There's one point. Randomish is a phrase I use. I like randomish. So there's the same chapter, still in chapter 4. There's a part where you say, X mechanism can't explain whatever, or X mechanism alone can't explain something. And that's always the case. No one mechanism can explain anything. It's always a combination of mechanisms. And if you break it out into like mechanisms 1, 2, and 3, you're never going to be able to explain anything. It's all about how they go together. In chapter 5, there's an example of what I call the gene for a leg problem, this was in the discussion of the IFT, the cilia protein factor, in the context of kidneys, and it has to do with the common idea that there's like a gene for legs and a gene, genes for eyes and genes for kidneys, and it doesn't work like that. There's genes that have patterns, right? So there's genes for folding and genes for extending, but there's no like gene for a leg. And it was so this specific thing with the kidneys was an example of that. And then in chapter 7, there was an example where for mutation rates, you described as per generation, but the way mutation rates are reported is per replication. So when you're talking about like chlamydomonas or plasmodium, then like it doesn't matter because they're single cellular. So they just, each generation is mitosis. But for humans, you have to account for all the rounds of mitosis from the zygote to the adult. So it's not a one-to-one thing. Now, those are all very small things. And I hope that didn't come away as mean or hostile or anything, because I just wanted to for the audience, just want them to know that like, there's a couple things here. I don't want to harp on them, but I just want people to be aware there's, just kind of small errors throughout the book.
Maybe I could just say something briefly, and that is,
please go ahead.
And many of the things that you point out, I'm not even quite sure what you're thinking about, but I'm happy to agree that when you write a book, sometimes you don't phrase things exactly, especially if you want it to be a broad readership. You can't have tedious technical details too much. So
that's fair.
I'm sure that some of the things that you point to could be said better.
No, that's definitely fair. And I struggle with that too, especially for like 100-level classes, stuff on YouTube. It's like, how do I be general without being incorrect? And it's often a very fine line. So I hear you on that one. I think some of that doesn't fall into that, but I think some of it could. So the first big thing I want to bring up is the broad picture of selection dynamics and the different ways that selection works. So as I said a few minutes ago, the examples that you employ here, it's plasmodium and how plasmodium interacts with humans. It's HIV, and then to a lesser extent the Lenski long-term evolution E. coli experiment. So do you remember? I don't know if you ever took like an evolution class as an undergraduate. Were you familiar with kind of the three, like a 100-level class, like the three broad modes of selection? Does that ring a bell at all?
I haven't come across it under that description. What did you have in mind?
So sorry, you can all, I don't know if people can hear my dog going in the background.
Yeah,
I apologize, everybody. My dog is losing his mind right now because there's
no problem.
People. Sorry, everybody. You're going to hear dog barking periodically. Okay, so so for when we look at selection, there's different ways that selection can operate on you have a distribution, and there's different ways selection can change that phenotypic distribution over generations.
Right.
So the situations that you focus on are all examples of directional selection, where you have a single strong selective pressure that is pushing a population in a specific direction. So for plasmodium and drug resistance, it's basically adapting to whatever dosages, right? It's the same kind of thing with antibiotic resistance. With HIV, it's adapting to a new host. With the Lenski long-term evolution experiment, it's adapting to the specific environment in that experiment. So you're imposing a set of selective pressures that are strong and they're constant, and the population is being shifted directionally, hence the name, directional selection, kind of to whatever more extreme to be more fit in that particular environment. I'm going to grab him in a second just so he can chill out in here with me so he stops yelling. So I apologize, everybody. I'm going to be right back.
I think he's cheering you on.
This is the perils of live TV, right? I'll be right back. All right, this is the offender right here. All right. Okay, so my apologies, everybody. So as I was saying, directional selection. So do you think, so my question is, sorry for the long lead, and my question is, do you think that directional selection is appropriate to be representative of a global maximum rate for how evolution can work?
Yeah, I do, because if there's not a particular selection pressure, which if it were not satisfied, say, then essentially one has pretty much neutral evolution. And again, one loses the magic of Darwin's mechanism of tiny changes followed by selection, tiny changes followed by selection. So yeah, if there is no selection pressure or positive selection pressure, then there is no nothing to push it in a particular direction, which these things would need.
So I agree, but there can be other ways of doing that. So so one of the other modes is called stabilizing selection, which is kind of just whatever the mean is, kind of preserve that, right? You take a distribution and you make it narrower and taller. But the opposite of that is disruptive selection, where you take a distribution and you make it wider and flatter. And ultimately, with strong disruptive selection, you end up with a bimodal distribution. And you often see that leading to speciation, or in extreme cases, adaptive radiation, where you have a single population that adapts to fill a bunch of different ecological niches. Why isn't that a better framework, or why isn't that a better representative in terms of a maximum rate of evolution?
Well,
you have more than one thing going on at a time there.
I think that is actually subsumed under the directional selection, because I forgot what the term is, bifurcating selection, disruptive selection. That's just being selected. There's several pressures, or the two peaks affect each other, and so they are under directional selection. But it's just that they are experiencing different selective pressures. So, for example, in my book, in the Edge of Evolution, talk about malaria in areas of the world which, say, didn't use chloroquine. Then the parasite didn't change too much, and in areas where it did, then it did evolve. But after it was all over, there was no particular change in the malaria. There was nothing cool arising in the resistant malaria, and nothing much changing in the non-resistant malaria.
Well, so I hear what you're saying, but you said a phrase that is exactly my point here. It's different selective pressures pushing it in different directions. And that's exactly what I mean, because what happens is you can get selection for different traits in subpopulations simultaneously. So whereas all of malaria, it's basically either stabilizing selection, where we're not treating it, it's basically stay the same, or treat it, and now you're being selected for drug resistance, right? But with disruptive selection, what you're doing is you're selecting for multiple different things at a time, within subgroups within a population. And a really cool example of this, just to make it concrete, is a species called apple maggot flies, which sounds gross and they look kind of gross, unless you're an entomologist, in which case I'm sure they look beautiful. But these are, so the ancestral species for apple maggot flies is hawthorn berries. They've adapted to apples as well. Apples are not native to North America, so they've adapted to apples once apples appeared here. And the interesting thing is these two plants fruit at different times of year. So what you're seeing is so this population used to have kind of a broad when they would reproduce over the time of year. As apple trees displace hawthorn bushes to some extent, you end up with kind of different times where those fruits are present. And you see the population bifurcating into ones that live just on the hawthorn berries and just on the apples. And what we're seeing is reproductive isolation. And we've actually looked into the genomes of these things and found that the two different populations are being selected differently from each other. So, and you can look at specific genes. It has to do with olfaction is part of it, where they have higher sensitivity to different compounds based on their food. But they're also seeing differences to the regulation around their reproductive timing, where they have to have their larvae emerge at different times of year to maximize the food that's present. My point here is that is happening just in the last couple of centuries, and it involves way more traits than drug resistance in Plasmodium. And the reason for that is you're selecting it simultaneously in two different directions. And so my first question here with this kind of big idea is, why aren't examples like that, where it's a population diversifying, specializing from multiple things, rather than specializing to one specific thing? Why aren't those diversification events better examples of a speed limit, if one exists, than the one-dimensional events?
Okay, they're perfectly fine examples of what Darwinian processes can do. As you mentioned, people looked at their genomes. I would need to know what the exact changes were at the molecular level to evaluate whether I thought they would or leading to some new molecular biochemical system or not. The nice thing about malaria is that we are able to get down to the amino acid level of proteins and see exactly what the mechanism is. I've stated a number of times, I forget where, but that in order to evaluate evolution, it's clear that Darwinian processes can do some things, but in order to evaluate evolution, you gotta look at the molecular level. You gotta see the mutations that are changing, because some things that impress us as big changes actually involve just smallish molecular changes. And on the other hand, some things that you wouldn't be too impressed by turn out to be very complex biochemical molecular systems. So I'm a biochemist, so that's where I focus my attention.
Wouldn't it I just want to clarify the argument in Edge of Evolution. It's the number that really matters, though, right? More than the nature of the changes. It's the number of specific amino acid substitutions required for a trait that's kind of the linchpin, right? I should say the number simultaneous.
Yes, that is correct.
Right.
And if they can't be formed sequentially, and they have to arise in several bunches at once, then that's the discontinuity that Darwin was worried about. Yeah.
So what I will say is, for the flies, they've documented positive selection, so selection for new substitutions across several genes, and they found that the hybrids between the two subgroups are lower fitness than either of the originals. So that implies that you can't do some of them. It's kind of an all or nothing thing. So I think that I think it fulfills the criteria of multiple things. You need them all at once, right? I think that fulfills it. And that rate is faster than Plasmodium based on the fly population size, the generation time, and the amount of time.
Well, I disagree. You know, I don't say that you can't select for this trait and that trait and the other trait and get multiple changes over time by a Darwinian process. I'm focused on things that, for say one trait, you would need multiple changes. I'm happy to agree that you could change, say, the timing of a fly's reproduction, or you could change the ability of it to metabolize a slightly different substrate for food and stuff. You could do those one at a time. But if it took the fly 2 amino acid changes in one protein to be able to take advantage of a new food source, that's where Darwin's mechanism gets into trouble.
So I think with the specific example of the flies, I think that works because they do need to, it does you no good if you're more receptive to different olfactory compounds if you reproduce at the wrong time to exploit that food source. So you do need to have multiple things. I do actually have one question. Then I want to move on to my next kind of big topic here. It's that why does it have to be in one protein? If there are 2 specific changes that are required, and we're talking, and the probability of one of them is 1 in 100 million, the probability of the other is 1 in 100, whatever it is.
Well,
why does it have to be the same protein?
It actually doesn't. I concentrate on that in The Edge of Evolution, and it's interesting because it can change a protein in steps. I talked about the protein PFCRT that is responsible for chloroquine resistance, and I also talk about some hemoglobin mutants that are derivatives of sickle hemoglobin to try and make it. But it's true that you could have, if you need a change in this one and a change in that one, yes, that would work too. As a matter of fact, your viewers might know that some drug cocktails that physicians subscribe for AIDS or other conditions contain multiple chemicals, multiple drugs that will block this protein or this enzyme and this one too, so that even if one mutation arises for one of them, the bugs will still die. So yeah, you can.
Yep. Great. So I'm gonna leave that piece there, talking about selection dynamics and specifically directional selection, and why I don't think directional selection is the best way to approach the question. I want to move on to something called the waiting time problem. Throughout the book, you talk about the difficulty of gaining 2 or more independent mutations, as you just said, 2 or more independent mutations that are all required for whatever trait it is that you want to select for, right? It's the difficulty of getting these 2 low probability events co-occurring in the same genome. You actually use the phrase waiting time in chapter 6. I don't know how familiar you are with the young Earth creationist literature. I'm extremely familiar with it, probably more so than I should be. And the waiting time problem is actually kind of a trope among young Earth creationists. It was actually kind of the flavor of the month back in like May or June. Like a bunch of people did like videos and wrote things about it all within a few weeks of each other from like Creation Ministries International and a couple other places. So don't make me guilty by association here. I don't read young Earth creationist sources. I didn't get the phrase from there, and maybe indirectly somebody I heard talking about it, but no, that's not my source for it. Yeah, I didn't think you did. As I was reading the chapter, I made notes of like, this sounds like the waiting time problem, and then on the next page you have the phrase waiting time in there, and I was like, oh,
I don't even remember that phrase coming up in the book.
I think that was convergent evolution, but there's jokes I can make there, but I'll put it aside. So the reason I bring this up is because the arguments you make by convergence are in substance the same. It's the waiting time problem, and it's the idea that if you need mutation A and you need mutation B, and each of them has whatever probability, the likelihood of getting A and B co-occurring in the same genome is very low. And then if you need additional mutations all at the same time, the probabilities compound. And as you describe it, it's an exponential increase in the difficulty, right? As you add.
Let me just say briefly that there is no need for convergence or some conspiratorial thing there. It's just that, hey, if we're going to get this from that, what's required? But how long would that take? It's a question that pops up pretty easily if you start to be skeptical about Darwinism.
So my question related to the waiting time problem, and I'm going to use the phrase "the waiting time problem," even though you didn't use that phrase. It's the same. It's the same. Fundamentally, it's the same argument. So I'm going to use the phrase.
Okay,
I know you didn't get it from there. I get it. So the question I have for you is, what do you think are the weaknesses of that argument?
Of the waiting time problem? Well,
Yeah.
of course, it's a probabilistic idea, because any mutations could arise at any time, since they're random. So it's kind of like saying, what would the odds be of pulling three numbers up in from an urn with numbers 1 to 100? You could calculate it, and it might be a long time or take a number of tries, but it could come up on the first or second or 16th try.
Right. So we don't. Sorry, I didn't mean to interrupt. Please continue.
Nonetheless, if you have to repeat that, the strong law of large numbers says that the average waiting time will certainly converge to the calculated average.
Gotcha. So for small numbers, it doesn't have to be the last one. If it takes X number of tries, it doesn't have to be X minus 1 or X. That's the answer. But as you get more and more to that, you converge on. It does actually take X number of tries to get the thing you're aiming for.
Right.
So the evolutionary problems with the waiting time problem, there's two big ones. One is the underlying assumption that that mutations occur in sequence rather than in parallel, right? Because actually, I should pause. I did get a super sticker from Raw Nakedness. Thank you. I appreciate you. Thank you for being here. You read the names. You know people are going to name. So thank you for that. I appreciate it. Back to the waiting time problem. So problem number one with the waiting time problem is that it treats evolution as an individual process that occurs in serial. It looks at one individual and says, okay, in this one genome, you get a mutation. And then how long does it take to get the next mutation and the next one and the next one all in a row? But evolution isn't an individual process that occurs serially like that. Evolution is a parallel process. Evolution occurs in populations. So everybody is experiencing mutations. So the probability is not of one specific person getting one specific mutation. It's the probability of that mutation occurring in the population as a whole, right? That's the baseline probability that you need to operate from. And then the second part of it, and I think the bigger problem with the waiting time problem, is that it ignores the phenomenon of recombination, where you have two mutations that occur in different genomes, and then the descendants of those mutants mate with each other or otherwise somehow exchange genetic information. And now you end up with those two mutations together. And there's a wealth of experimental and observational evidence that shows how recombination processes, whether it's sexual reproduction or just other, horizontal gene transfer, whatever it is, how those processes greatly decrease the time it takes to get two independent mutations simultaneously.
Well, I think that's ignoring the kind of stipulation I made that the individual mutations don't help or hurt, that you need multiple mutations at once. It's true that populations can vary in all their positions at any time, but at any time it's unlikely to have a particular mutation, if it is a neutral one, in a large fraction of the population. So I've seen calculations, and it does affect things a little bit, but ultimately it doesn't affect them enough to save Darwinian processes.
So I have two things. So one is just numbers, just looking at numbers. Humans, for example, there's about 7 billion, now 7.5. There's some 7 billion and change humans on the planet. We have our haploid genome is 3 billion bases, and we get about 100 mutations per person per generation that are new, right? Now, if you do the math on that, you can just do the simple math. 7 billion times 100 is 700 billion, and the diploid genome is only 6 billion. So within the population that exists right now, we've sampled every non-fatal single base substitution many times over within the human population right now. And we have a relatively small population and a relatively huge genome compared to lots of other things. So when you get smaller scales with larger populations and smaller genomes, that frequency problem goes away really fast in terms of, I mean, you're not talking 1 or 2 or 5%, but you're not talking 0.000, you know, with 10 zeros, right? It's several orders of magnitude different for many, many, many things in terms of that frequency. And once you hit a certain frequency, if it's neutral, it's the likelihood of fixation is approximately that frequency. So you just have to hang out long enough for the second one to appear. You don't have to be selected for. And the more conceptually, though, so we can talk about the numbers if you want. Go ahead, but there's a conceptual problem that I want to hone in on here.
Okay. Well, one can ask a person with your position, so in our population, what multi-mutation event has come along to help, say, with malaria, with folks in malarious regions? There have been a number, there's actually only a relative handful of kinds of mutations. This bug is deadly. It can eliminate humans in some regions. There's intense selective pressure, and yet there's only a handful of mutations that can help, and none of them are coordinated with each other in a strong sense. None of them are on their way to building anything coherent. You've got the sickle cell mutation, that's one of the better ones. And you've got a number of broken genes, globin genes that lead to thalassemia, glucose 6-phosphate dehydrogenase deficiency, all sorts of things like that. One problem with folks who talk about think that unintelligent or undirected processes do all the work in evolution is that they tend to, if there isn't data to constrain them, they tend to, say, well, if I got this one and then got that one, and they're actually using their intelligence to try to map out a path to get where they want to go. But there's no indication that nature will take that path. And so that's a real danger. So that's why one should rely more on experimental or field results than on one's models.
So I hear you, but that's the very common fallacy of having a specific target, which evolution does not have. And you use the phrase where they want to go. Evolution doesn't want to go anywhere. Evolution doesn't care, if it's in this gene for, hemoglobin or one of the other ones that actually you don't mention in the book is platelet. I always forget it was platelet factor 4, platelet factor 6. There's one of them that is actually affirmatively like anti-malarial, or some variants of it are affirmatively anti-malarial. It's not breaking anything. It's a whole different thing, but that's neither here nor there. The conceptual point I want to make on this is that if recombination was not a solution to the waiting time problem, then we don't have a good explanation for why sexual reproduction is so common. And when asexual and sexual populations compete, almost always the sexual ones win. Like we see asexuality evolving in animals. They go from sexual to asexual, but there are very few examples of ancient asexual mammals because they lose, because doing that recombination is so beneficial, both in terms of getting mutations together and separating out mutations. So you can take, if you have a good one that's linked to a harmful one, you could separate those apart. If those benefits didn't exist, we don't have a good explanation for why, like, we go through all the trouble of doing sexual reproduction.
Well, yeah. Well, let me just point out that you say we don't have a good explanation for why it exists. We look back and say, hey, this is helpful, but we don't know why. Or we think that according to our calculations, it'd be more beneficial to be asexual because you get more of your genes into the next generation, although it's a whole lot less fun. And let me just point out for the listeners that The origin of sexual reproduction, as a matter of fact, the existence of sexual reproduction, has been a mystery for evolution for 100 plus years.
Well, I want to point out that that one we have a pretty good handle on how that one.
Yeah,
we got that one.
Okay,
we got a pretty good handle on that.
I could cite that, and it's kind of like, well, no, let me go to a different topic. I'll leave that there. And you said that we don't have a goal, and I agree that evolution doesn't have a goal, but evolutionary biologists do. And what they want to do is say, hey, how can I explain this? I know if I did this, that, and the other thing, or if this happened in nature, I would end up with it. But, that's using goal-directed thinking to try to explain something that you've got. As a matter of fact, that's what was done here with sexual reproduction. You say it's good, so it must have evolved somehow. But let me point out that in nature, in an astronomical number of attempts, malaria didn't develop anything. It was free to try whatever pathway. We weren't directing it. It could come up with any counter to chloroquine that it could manage. And in 10 to the 20th or so attempts, it came up with two or so amino acid changes in a particular protein. And if you think about the implications, that means that there were no effective answers to chloroquine that had a frequency of occurrence higher than 1 in 10 to the 20th. So not only have we got that number, we have eliminated things that are more probable than what occurred.
I don't want to get bogged down in the numbers here. That last claim is wrong for a very specific reason, and it has to do with the selection dynamics that appear in a population once one or more beneficial mutations has appeared. I don't want to go down that rabbit hole because there are other big topics I want to talk about, but I just want to note that you can't, so treating this as a probability game once selection enters it doesn't work. And that's fundamentally, that's the problem there. And then that tangentially related to the recombination problem with the waiting time. So the next big topic I want to talk about is fitness landscapes. This was a big thing in chapter 6. The idea being, and I'll kind of summarize this and tell me, feel free to adjust it, tell me if I'm missing anything. But the idea is that if you have a Darwinian process, right, you're increasing benefit or functionality or whatever from a low fitness to a high fitness phenotype. You could treat this like a three-dimensional landscape. And the problem is that it's not a smooth landscape where you have one smooth slope all the way around a high peak, right? Instead, it's a big craggy landscape. You got hills and valleys and mountains and canyons. And if you get stuck on a local plateau and every change from that site makes you worse, then there's no way to get to the high peak. And you could use, geographic examples. If you get stuck on Mount Kilimanjaro, there's no Darwinian pathway to Mount Everest, right? Because you have to go down in every direction in order to get to that higher peak. Is that a fair representation of the fitness landscape argument?
Good.
So that only works if fitness landscapes are constant. If a given genotype has a universal fitness cost or fitness effect across the board of ecological conditions, that it stays the same indefinitely. If you get onto a local fitness peak that's not the global fitness peak, you're only stuck there if that local peak is always a local peak relative to the global maximum. If it becomes a valley, if it merges with the maximum, then it's no longer a problem. Is it the case that fitness landscapes are constant?
I don't think so, but they do not vary in ways that would lead from something simple to a coherent piece of machinery. The problem with talking about that, fitness landscapes can vary, but folks have seen that if they do vary and vary too frequently, then the organism or the expected mutations kind of wash out, because at first a species is trying to adapt to this, and then over here, and then over here. Additionally, if you're trying to explain molecular machinery, such as I focus on in my books, like say, oh, the bacterial flagellum, that is, the poster child here, you would have to think that there was a wonderfully coherent or even maybe designed change in the fitness landscape to have mutations that were being selected for individual changes in environment to add up to something that is an integrated standalone system.
So I think putting aside kind of the difficulty of quantifying that, there's experimental evidence that what you just said isn't the case. The example that I can think of off the top of my head is looking at, I think it was, I'm going to get the virus wrong. I think it was vesicular stomatitis virus. It was some human virus. And it was adapted on both HeLa cells, which are human cells, and MDCK cells, which are canine cells. I think they're canine kidney cells. And in this experiment, you have your relative fitness, right, of this virus in each one. You just set the relative fitness to 1, and you can adapt it to the human cells, and its relative fitness shoots up to like 4, 5, 6, like it's really good, but it loses out on the dog cells, right? It goes down to like 0.5, whatever. Mm-hmm. You go the other way too. You could adapt it to the canine cells. It gets really good at infecting the canine cells, but it has a really hard time on the human cells. The other thing you can do is you can switch back and forth every day between the two types of cells, right? So you could flip the fitness landscape back and forth between the two environments, or you can do it randomly. You can flip a coin each day and decide what type of cell you're going to be on. In those two latter cases, you see an increase in relative fitness on both types of cells that are equivalent to the specialists for each kind. So you have a generalist that actually maximizes its fitness in both fitness landscapes, that it's an even competitor with the specialist for each one. And that's changing your fitness landscape, getting you, there's obviously valleys between those two peaks, as is indicated by the specialists. But by just changing it back and forth, you generate generalists that are as good.
That's fine, but I don't think we're talking about the same thing. I'm talking about coherent molecular machinery or proteins or so on. Mm-hmm. I don't know the particulars of the example that you're thinking about, but I would imagine that they're binding to homologous proteins in the two different cells of the two different species, and they mutate between one and the other. Viral mutation rates are very high. I wouldn't classify such a change as much at all. And I think a much better example of what happens for a microorganism adapting to an environment is Lenski's experiments. They have lots of data over lots of generations down to the nucleotide level. And as I've written, I'm sure you've read, they see nothing that indicates that the bug is developing any substantially new information-bearing system. It's mostly degradation.
So two things here. One is that over the course of that explanation about just the viruses and the two types of cells, it went from, I don't know the specifics of what it is, to it's no big deal. It can't be both of those things. Either you know exactly what it is, right? In which case we can characterize it, or you don't know what specific it is. So you can't say both. I don't know the specifics of that example, but also it's no big deal and it's not any new biochemical machinery. It has to be one or the other, right?
Well, no, I said that if it were as I expect it to be, that is the same protein from the virus hooking onto a homologous protein from this species and that species, then I would not think it to be a big deal. If there were a new protein or even a different protein from the virus that developed a different a binding site to a different protein between the two species, then I would be more impressed.
So,
As
I mean, we have examples of that too, though. I mean, I said at the top I wasn't going to rehash the HIV example, but the example of a novel binding site.
I have tried to track them down, and people have sent me things, and I have to admit that when I look at the details, they're not talking about the same thing that I am.
So I will say with the HIV example, it's not a complicated example. It's the transmembrane domain of the VPU protein. It evolved a new binding site for a human protein called tetherin that involves at least 4, as many as 7 specific amino acid substitutions. And in substitution experiments, it is an irreducible trait. I said at the top, we're not going to go down I don't want to go down the rabbit hole of the HIV example because it's been hashed out to death, but it does fulfill the thing you're saying.
Let me just say for your viewers that I've heard the exact exception that you're talking about, and I've answered it, showed why it is not responsive to what I wrote in Edge of Evolution. I said specifically in Edge of Evolution, I was talking about cellular protein binding sites, and I specifically excluded viral proteins from my analysis. And one of the things that I run into is that people don't read these things with much attention. I'm trying to make distinctions here, and I find that some readers of my work don't notice the distinctions and therefore fail to understand the argument.
So that seems like a very arbitrary distinction. If the argument is that globally there's a speed limit, you can't get a trait that involves more than X number of specific substitutions. We have an example. Well, HIV doesn't really count. I will say though, that back when you were doing this kind of blog back and forth with Abbie Smith, Dr. Abbie Smith and Dr. Ian Musgrave, you did ultimately acknowledge that this was an example of novel biochemical evolution in there. Like you did acknowledge that HIV example did fulfill your criteria. That was back in, I want to say 2008, 2009, somewhere in there. You did ultimately acknowledge that it did.
That's right. But once more, in the book itself, which was published in 2007, I said I'm talking about protein binding sites. The problem with viruses is that they can mutate a whole lot more rapidly than cells, that they have a whole landscape of targets that they can try to mess up. And so the numbers are simply different for them. So if you're going to say, well, we're not going to put any rate limits on this, then you're fine, but you're leaving biology behind, or at least the aspect of biology that I'm talking about.
So I want to move on to this because I do have one more big point I want to cover before we wrap it up. But I do want to say that if we acknowledge that like the HIV example is a protein binding site, it does have 4 to 7 specific substitutions that are required for it. And in the book, you specifically use HIV as an example of the speed limit. You say, look at this huge population of viruses, even within an individual person, right? I forget if it was, I think it's trillions is the population size globally. And
that's right.
Right. And you actually say, like, look, even HIV with this enormous population can't accomplish anything. So how can we say any of this other stuff? But the fact is that HIV did do exactly that. And so whatever the top of that distribution is, you just lop off like 20 orders of magnitude from those numbers.
I disagree.
Oh, and there goes the dog again.
Let me just say briefly, I know that you want to get to a different point, but I specifically pointed out that I said HIV had no protein binding sites, but yes, in conversation with this lady,
with this doctor, doctor.
Now she is. Then she wasn't, I agreed that one had developed. Okay, but if you remember in chapter, I think it's 6 or 7, there's the title of the chapter is called "The 2 Binding Site Rule," and I said that the edge of evolution is 2 binding sites, and that counting malaria, counting HIV, none of them crossed that threshold that I discussed. So I know you don't want to get into the weeds here.
Yeah, we won't get into the weeds a bit, but I disagree.
Just want to let the viewers know that I disagree.
That's actually a really nice lead in, the two binding site rule, because the very last thing I wanted to talk about before, I have one small question to ask you at the end if we have time. We'll see how we're feeling, but there's one kind of big idea I wanted to get to before we kind of wind down, and it's the idea of this overall approach of looking at well-documented examples of molecular evolution and kind of working forward from there, saying, okay, given huge population size over whatever amount of time, here are the specific things we've seen, what traits did that result in, and then use that as representative of a speed limit. And I hope you're okay with me using the phrase speed limit. I don't think it appears in the book, but yeah. I've been in my head just kind of speed limit. Yeah. So what I want to ask you about is, if the goal is to establish what traits cannot evolve, what, as we start at the top, we agree, universal common ancestry, we want to know what along the way we cannot attribute to evolution. So wouldn't a better approach be to start with the traits and to look at what traits we can witness evolving, and then dig into the molecular genetics of those traits and say, okay, given that we witnessed this trait evolving either in nature or in a lab, we can say that that is under the ceiling, right? That is within the speed limit, whatever that speed limit is. If we can observe it happening in real time, then that's within the speed limit. And the reason I ask this is because I think we have a bunch of examples that violate the two binding site rule that you articulate. And the two that I really like off the top of my head are the multiple examples of endosymbiosis that are going on right now. One example of primary endosymbiosis, and then a dozen more of secondary endosymbiosis within animals, which is really cool. The primary is in a rhizarian, Pollinella. The secondary, there's a bunch of animals that are basically becoming photosynthetic right now. And then the other example is placentas evolving in lizards. We've witnessed a population of lizards evolving true placentas. And now, in order to have a placenta, right, and this is not just oviviparity. It's not that the eggs develop internally without a shell. It's true uterus placenta interface with the blood of the parent and the embryo. And in order to have that evolve, you need way more than two distinct binding sites, right? You need to regulate the growth of the blood vessels. You need to have the interface of the blood vessels. You need to have tissues that are receptive to the embryo. You need the embryo to suppress the parent's immune system. And if any of those things don't work, if you don't have any of those specific interactions that are perfectly calibrated, then that's not going to evolve. But this is a population of lizards that, like, they didn't have placentas, and now they do.
Well, I would disagree. In order to understand what evolution is doing, in particular whether Darwinian processes can explain what you see, you gotta go to the molecular level. In the case of the lizards you're talking about, I don't know, maybe there is molecular level data, but in the examples that I am familiar with, there is no such processes taking place. Again, if you look at microbes like Plasmodium that occurs in large numbers, and which was the focus of the book, it does not do anything fancy at all. And Richard Lenski's...
if I can, I apologize for interrupting, but if I can, this example is doing something very fancy. So you said you disagree. Do you disagree that it's placentas? Do you disagree that that's a complex trait requiring a bunch of interactions? Like, what specifically do you disagree with here?
I strongly suspect that there is no novel molecular machinery or binding sites, or what I called in my quarterly review of biology article in 2010, functional coded elements. That is, new transcription factor sites, new protein modification sites, and so on. In the case of the lizards that are developing something new, it might be that maybe there was already a fair amount of genetic information there, maybe, it devolved. Maybe some of these ideas might not be as likely as others, but you gotta look at the organism, the DNA level, the molecular level. Otherwise, you're essentially stuck in your imagination.
So, okay, so do you think it's possible then to evolve placentas from an ancestral egg-laying state without any, two new binding sites? Because if that's the case, then my question is, what morphological complexity is not possible? If you could get something as complex as placental development with nothing new.
Okay, here's something. Suppose you had an organism, maybe not the ones you got, that had, say, the ability to bear placental organisms and lost that trait. And then later on, a field biologist goes and sees that some descendants of these are placental producing organisms. But it turns out that a single change is necessary to reinstate a pre-existing path. In that case, one's amazement at the discovery of an organism with this feature would be mistaken, because almost all of the information for doing that was already there.
There are some examples of that. So, for example, you can induce multicellularity in Saccharomyces cerevisiae, for example, right? That's a fungus. It's yeast.
Yeah, I've read that. It's a very
the ancestral state. The ancestral state is multicellular, and it's lost it, but it has all the genetic machinery there to become multicellular. You can induce it in the lab, right? But that's not the case for placentas, and it's certainly not the case for photosynthetic organelles in animals, right? I mean, right? We know what the common ancestors of mammals and reptiles were, and they were not giving live birth.
I'm not familiar with the examples you're citing. I would be delighted to read about them. If you've got citations, I'd appreciate it if you could send it on to me. But based on what I have seen, I am very skeptical.
Fair, but I assure you, I'm not pulling anyone's leg here. We have witnessed the evolution of placentas and lizards. We have witnessed animals, like there are animals right now that transmit photosynthetic organelles through their eggs. Like you can look at the eggs and they're green. And there are other animals that actually have photosynthetic genes, in at least one, in their nuclear DNA.
So all you're talking about is common descent. You're not talking about how that happens. You're not talking about, you're saying, look, there's an organelle here and these have this over there. But Darwin's big idea was not just evolution or common descent. It was random mutation and natural selection.
So that's exactly my point, though. It's not, for these examples, they're not explainable via common descent. Reptiles, like a specific lineage of lizards that has placentas, to the exclusion of all other extant reptiles. Like none of them have placentas, just this one group of lizards, right? That's a new thing. It's not common descent.
It's also the case that if a Darwinist would be startled and would never predict a lizard to eventually get a placenta. So even if that were the case, it doesn't fit into Darwinian theory very comfortably.
That actually isn't the case.
I don't know the details of it. So
it actually isn't the case, because people were looking for it, because we have lizards that are oviparous, right? They lay eggs. That's what most reptiles do. There are lizards that are oviviviparous, where the eggs develop internally. And it's hypothesized that that's a step on the way to in utero development. So people were looking at these oviviviparous lizards and saying, hey, I wonder if any of them are further down that line. People did explicitly predict that you might find placentas in there. And lo and behold, we did.
Okay. One more time, though, that this is just about common descent. Did some people say that, if you switch this gene, if you change that gene, and so on, we would lead to this new tissue? If you think, as some people do, that evolution is directed, that information was added along the way or some such thing, then it's not does not follow that just because you see new things occurring or developing, that it was because of Darwin's mechanism of random changes and natural selection.
And that's actually not a bad place to move on, because that's kind of my point, is you don't need to know the mechanism. If you can say, here's a thing that's new that evolved, here's a trait that's new that evolved, we don't have to wonder if that trait can evolve. We can like witness it. We can see it happening, right? We can like watch.
The question is how? How?
Well, right. Okay, so are you proposing then that the placentas are designed?
Could be.
Because okay, so they can be, so the design can be an ongoing thing. Is that?
Yeah, I have nothing to rule that out. So
okay.
What's the evidence that it occurred by random mutation and selection?
That we can watch it happen and document in real time the changes as the population, and then we can look at differences in the genome between the different populations and see different genes, different gene expression pattern. Like you can do this, for example, using the animal example with the endosymbiotic. They steal the plastids from some green algae that they eat. We can actually document the presence of a gene for photosynthesis in the nuclear genome. The process is called endosymbiont gene transfer, where you shrink the genome of the endosymbiont and bring it into the nucleus. We can watch that happening in 2 different species right now in real time.
Well, that's great. I don't deny that something like that could occur, but the gene was there in the first place. So the only difference is that the gene was copied, inserted into the genome for a different species.
So that's a different question, though. The origin of those genes is a completely different question for how we get complex traits in the present. Now, I do want to point out here that there's a bit of a double standard where I'm being asked to explain the mechanism in detail, step by step. But if I was to say, what's the mechanism of design? When did it happen? Through what process did it happen? And how can we document that process? I don't suspect detailed answers will be forthcoming.
That's because Darwinism claims to be the mechanism. And so a criticism of Darwinism would say, well, the mechanism that you propose is grossly inadequate to explain these features of life. On the other hand, intelligent design is not a mechanism. Intelligence is not a mechanism. And rather, it is identified by other criteria, which I discuss at length, or I've discussed a number of times and at length in my recent book, Darwin Devolves, that when we see purposeful arrangement of parts, we always understand that to be the result of design. And that, in fact, is a basic facet of human reasoning to understand that, how do we recognize other minds? How do we recognize intelligence? And may I say that the lack of a mechanism is not an obstacle to a scientific theory. For example, Isaac Newton had no mechanism to propose when he talked about gravity. No mechanism is known that could produce the universe. And yet the Big Bang is widely accepted. Gravity is widely accepted, and there are other examples too. So the fact that you can identify a pattern, such as say planets and galaxies moving away from each other as if in the aftermath of a big explosion, and propose that the universe started in an explosion, even if you don't know quite how, that doesn't mean it's not scientific. In the same way, if you see a machine put together, you say, oh, that's the result of intelligent agency. And we can say that either in our own lives when we see a mousetrap, or we can say it when we see machinery in cells.
Okay, so I just want to be clear before we continue that we're not positing a mechanism for design. We're just identifying cases of design. We're not making an attempt to posit a mechanism. We're just saying, if you meet the criteria, design. So just to wrap up that last segment there, the idea I'm trying to get at for everybody is just that if you can identify traits as evolving, like you can witness them evolving, then the underlying biomolecular genetics is almost immaterial if you can get things as complex as endosymbiosis or multicellularity or whatever, all these things we can directly observe, placentas. If you can directly observe them, then you don't have to wonder if those things can evolve. That's the point. So we are getting.
You do have to wonder. You do have to wonder how in the world that's happening. If you saw Dr. Jekyll turning into Mr. Hyde, you wouldn't have to worry if that could happen, because it did. But you might really be curious as to what could make that happen.
So the argument you're putting forward in the book is that they can't. Right, is certain things are beyond the edge of evolution, that they can't evolve. And
the edge of Darwinian evolution, unintelligent evolution.
That goes back to I think the very first thing I said, which is like I don't adhere to Darwinian evolution. That's not a thing in 2021 anymore. There's a bunch of process, but we don't need to go down that road. So
let me just say, I disagree. And I think the default explanation is Darwinian. And I can go into that.
You're not arguing with me anymore. Now you're arguing with the dictionary at this point. Like, that's not what the word means.
Well, I disagree. Mm-hmm.
Okay. So the last thing, and I do want to thank you. It's now almost 2:15, so I do want to bring this home. I do want to thank you for being extremely generous with your time. We've got a little bit over here. So the last thing I want to ask, and this is just aside from the contents of the book, is I want to ask about the process of the book itself. So you may kind of notice that and people watching may notice that in kind of going through this, I'm kind of approaching this like a peer reviewer might, right? You go through this, you say, okay, have the methods been done correctly? Is the math correct? Did you do a thorough literature review? Those are questions that a lot of people that have looked at Edge of Evolution have addressed in a ton of detail. And I've got some links in the video description for a lot of the writing on that. The questions that I hope that we talked about, and I think we talked about in quite a bit of detail, are the methods appropriate to the question of like, where is the limit of what evolution can accomplish? And is the underlying theoretical basis for the work strong and or valid? And reading the book, coming away, the answer that I come away with for both of those questions is no. So I just want to ask you, at any point in the process, did an evolutionary biologist review the book before publication?
Oh, I have a number of friends who have expertise in a number of biology disciplines. I sent it out to a number of people. I wanted to send it to some evolutionary biologists that were better known, and they refused to read it. And I should say that my book, did not have a pre-review, but it's had lots and lots of post reviews, as you know. And when you link to the articles that you said you were going to, I hope you link to my responses to some of the reviews.
Yep. The whole threads. Yep.
Great.
I got the whole threads linked one after the other. Yeah.
Because I think that virtually all the reviews were bluster and bluff, and I've got specifics in the reviews.
Well, except for the one that ultimately agreed with HIV.
Well, yeah, okay, but again, I think that is not quite on target.
So if you're writing a book about evolution, it has evolution in the title, right? Do you think it's a problem that there weren't any like working evolutionary biologists that like reviewed it beforehand just to check the underlying theory? Like the stuff that I pointed out at the beginning about like just the units for mutation rate, for example, just like, do you think it's a problem? Do you think it detracts from the work that it didn't get a look like that before it was published?
Well, I would have liked it to, but again, as I said, a lot of people refused to do so. I sent out, or my publisher sent out, the manuscript for Darwin's Black Box before it was published. It had a number of reviews, many of which were semi-positive, but none of which could address the problems I raised. And I have seen reviews by not just a graduate student, but there have been reviews by Jerry Coyne, a noted evolutionary biologist, Richard Dawkins, Kenneth Miller, and Sean Carroll, in their reviews, they have nothing to say about that. I think touches the argument in Edge of Evolution. And that's too involved, of course, to go into here, but I say those things in the replies to their reviews.
I do want to be clear, though, those are critical reviews. Those were not like manuscript peer reviews, just to be clear. Okay, yeah, that's what I was asking. I wanted to be just to be clear on that one. Thank you. All right. So I don't have anything else to add to that, given the time that we're at. It's a little after 2:15 here. So, Dr. Behe, I want to thank you again for being so generous with your time and coming on. Just at the end there, you mentioned Darwin Devolves, and I'll email you. I would love to have one more conversation because we'll just go book by book.
Sure.
I'd love to do one more, talk about Darwin Devolves, because we've actually been poking around the edges of the Lenski experiment for both of our conversations. And like, I would love to go into, if I recall, that's a fairly significant part of Darwin Devolves. I would love to talk about that. If you would be open to that. So I'll email you and we can maybe do that in the future.
That'd be great.
Everybody, I hope you found that enjoyable and informative. This has been Dr. Michael Behe with me to talk about the Edge of Evolution. Thank you all for being here. I hope you have a great day. And if you are anywhere where it's really hot right now, stay cool because, man, it's really bad right now. Thanks for watching, everybody. Have a good day.